Thank you very much for that lovely introduction. So today we're going to be presenting on a topic, as you guys can see here, titled Urokinase-type Plasminogen Activator Receptor. Hopefully throughout that presentation, we won't be saying that long title anymore. We'll be just referring to it as uPAR. And it's a unique inflammatory pathway activator in many diseases. We stumbled upon this project. Student Doctor Mostafa recently got the opportunity to work with Dr. Lucas, who is the PI on our paper over the summer. And during this year, we noticed that there was a gap for uPAR in the literature in terms of its functionality, as more and more recent studies have been coming out about it. So we decided to bring together this review that focuses on uPAR, its functions in different pathologies, and some unique treatments that are currently trying to target it. So Student Doctor Mostafa will start off this presentation describing to you guys what is uPAR.
Hello, everyone. Hello, everyone. So my name is Student Doctor Mostafa, and thanks, Omer, for passing it on to me. So as Omer mentioned, the uPAR is a is a receptor. It's a receptor protein that is found on a variety number of cells in various number of tissues, ranging from skeletal tissue, soft tissue, myocardial tissue, and smooth muscle tissue, like all over throughout the body, which makes it a very interesting receptor to sort of analyze and, like Omer said, delve deep into the literature and see what kind of correlations can this receptor lead us to in terms of cell inflammation, intracellular versus extracellular. So now that we know that uPAR has been extensively studied, we show that these interactions of uPAR with cellular integrins, the GPIs and GPCRs, and that leads to a variety of intracellular and extracellular systems that it modulates. So, for example, Omer will talk about this later, which is the survival and metastasis in cancer, and how uPAR plays a factor in that. And I will be talking about the cardiovascular aspect of it. So the fibrinolysis, the remodeling, and as well as the complement activation. So uPAR is pretty significant, as I mentioned. It has a lot of different abilities to interact with a variety of systems. So it can be used as a marker for inflammation, a marker for cell cycles, a marker for migration of cells. And this process is very tightly regulated by a variety of different proteins on the cell surface. And it involves all the domains of uPAR, which is very unique for cell receptor, where only, for example, the extracellular domain would be interacting. And this interaction of all the domains in uPAR has the ability to cleave uPAR or urokinase plasminogen activator. Like I mentioned before, uPAR is a cell membrane receptor that is not directly attached to the cell membrane, but in fact it does go through a GPI, which is a protein, phospholipid C specific protein. And through this interaction, this protein GPI is able to be cleaved, which releases uPAR into a fragment called suPAR, or soluble uPAR. And that fragment is able to be detected in the blood or in the serum as a marker for inflammation in a variety of different cases. So that again delves deep into the factor of why is it that this factor is circulating in the blood. Does it have a correlation with inflammation, with cell migration, et cetera, et cetera. And this suPAR interaction is a key player in tumor and cancer progression as a marker for disease and a marker for inflammation, as well as other pathogenesis. So this is also a schematic that we use in our paper of how the GPI protein is being cleaved. So given that its cleavage site is on the cell membrane, you can have a suPAR or soluble uPAR element fragment that is free floating in the blood that can be detected through antibodies in the blood serum. And that could, like we'll talk about in a few slides, that can lead to an idea of the inflammation state of the body, the immune activation system, the malignancies, thrombosis, and other intracellular signaling that may be going on that can give us an indication of the disease states or the progression or the prognosis as well.
Okay, so next, as Mustafa just showed, that GPI linker linked to the membrane. So before we dive into the more of the pathologies of uPAR, in order for uPAR to play that really important role that we've been really hyping up all this time and all the biochemical pathways, we got to understand a little bit about these regions on the cell membrane that we call lipid rafts. And these are just areas of thickened plasma membrane that usually have slightly different composition to the areas around them, and they allow for more binding of a variety of proteins, and they also allow for the close proximity of these proteins, such as, in this case, uPAR, along with a few of its other members. And this is another picture from our review that we made, and this one is focuses on that lipid raft idea that I just mentioned and how it allows uPAR to be in proximity with the GPCRs and integrins. And later throughout this paper, you would see why these close associations in this little lipid raft are so important for it to promote proper function. This also offers another avenue in terms of detection of uPAR on the cell membrane as areas that have increased lipid variety compared to the normal plasma membrane can be detected through different ways of microscopy, through different lipid detection methods. So that will be very interesting, and we'll describe a little bit more about that coming up. So first of all, Mustafa is going to talk about the significance of uPAR in cardiovascular medicine, and then I'll follow up with its significance in oncology.
Oh, thank you, Omer. So as Omer mentioned, these are two sort of samples that we have taken from the paper, from the review article. First is the cardiovascular medicine and the oncology aspect of it. But uPAR is in fact involved in many, many different ways. For example, wound healing, for example, the GI system as well, which I encourage everyone to look into our paper when it's published by the end of this week, so that they can get a full appreciation of what uPAR is involved in. But I would want to talk about three different aspects of cardiovascular medicine where uPAR can be applied and has been applied in modern literature. So as you know, as you may or may not know, this gold standard of care for myocardial infarctions, infarctions, and specifically STEMIs, is a PCI, which is percutaneous coronary intervention. And in the past, what physicians have been doing in order to intervene in a STEMI is to perform a PCI as well as fibrinolysis. And there has been research showing the efficacy of using fibrinolysis along with a PCI. Initially, back in 2010, there has been more research onto using pro-uPAR, not the receptor, but uPAR as a plasminogen activator to activate plasmin and to induce fibrinolysis. However, this medication has not yet been approved by FDA. But however, research has now taken into a different avenue, a different lane in this field, where there has been a correlation between elevated plasma uPAR concentrations, or soluble uPAR, with the severity of cardiovascular diseases. So as you can see in this figure, there is a very positive correlation between patients with an elevated uPAR levels and the prediction of a future adverse effect of the cardiac events. And the pathogenesis, or the analysis of this finding, indicates that a rise in uPAR levels mirrors the release of inflammatory cells in the event of an MI. And this potential finding of measuring the patient's uPAR levels, for example, can help guide therapy into thinking, would the patient outcome be more favorable if it was through a fibrinolytic method or a PCI or angioplasty method. So this is something really interesting that is currently evolving, and it's taking more shape in cardiovascular medicine and its application to uPAR. It's not moving. Okay. Um, another aspect of cardiovascular medicine is an acute ischemic stroke, and here there has been many extensive trials that have looked at uPAR as a potential therapy for acute strokes. And here we can see the suPAR being used as a diagnostic indicator for unstable disease progression, specifically in stroke and hemorrhagic stroke, as well as ischemic stroke. So by analyzing levels of local suPAR in the bloodstream at the artery of infarction, it can provide many insights into the physiological role of these inflammatory cells proliferating and dividing in the area of stroke. And that could help also physicians and clinicians guide the care and of guide the care of the patients. And this figure and this table also shows the higher correlation between hemorrhagic stroke as well as ischemic stroke with the levels of pro suPAR. A third and final aspect that I want to cover today in today's short presentation, as much as I would want to delve deeper and deeper into the significance of uPAR and how interesting it is in the development of medicine lately, is angiogenesis. And from what we can see from modern research coming out, is that uPAR is expressed on the surface of vascular smooth muscles, which induces an intricate intercellular signaling cascade, which ultimately results in migration of cells into an area of proliferation, which Omer will be talking about in the setting of an oncology. And then looking deeper into this angiogenesis pathway and cascade, we can see that uPAR binding, uPAR and uPAR binding can activate the JAK-STAT kinase pathway, which we know from learning about it in med school currently, which then further elevates the level of plasmid-derived growth factor receptors, and then that drives smooth muscle proliferation. And in this setting where we can see angiogenesis, there is an overlap between the activity of angiogenesis in a specific tissue and uPAR levels. And as you can see on the right, there's an overlap in fluoroscopy between the uPAR levels as well as the platelet-derived growth factor receptor. And that is a correlation which has never been discovered before with a cell surface receptor, and which makes it a very interesting receptor to delve deep into and understand more of what it means to have it present in the areas of angiogenesis. And as you can see on the left, studies have also shown that there is an increased levels of, there's an impaired levels of VEGF-mediated signaling when uPAR is knocked down. So VEGF-mediated signaling is also this is specifically in umbilical vein and endothelial cell lines for both of these studies, just to be clear. And this knockdown of uPAR has shown that there's an impaired level of angiogenesis, which again stresses the fact and the importance of uPAR in the angiogenesis pathway, and this could be further applicable in medicine, where we assess the patient's ability to heal or ability to provide new vascularization to dead tissue and aquatic tissue following a brain bleed, following a brain injury, and following a myocardial infarction. And this is also further explanation to the human umbilical vein endothelial cells, where the knockdown of suPAR at the gene level has driven down angiogenesis to a very significant extent.
Okay, so next I'll be taking it over a slight different path, which shares a lot of similarities to what Mostafa has been talking about. But more recently, uPAR has been making a big appearance in the cancer world, and 3 cancers that I'll be highlighting today with most evidence of uPAR in them will be breast, lung, and prostate cancer. So diving right into it. So first and the most prevalent type of cancer where uPAR is found is HER-positive cancer. And as you know, it's well-known, highly aggressive metastatic cancer in nature, though curable, as we know, through molecular targeting therapies. The interest of analyzing uPAR in breast cancer stems from those few patients that are chemo-resistant to this molecular targeted therapy that targets HER2. So the question arises, is there any other factors that can help determine if this HER2 positive breast cancer will not respond to molecular targeted therapy in different groups of people? So recent work by this group came up with this hypothesis that HER2 and uPAR both work as co-amplifications of each other. So if HER2 is available as well as an upregulation of uPAR, this kind of causes a domino effect where it increases the numbers of these proteins quite high and compared to just one being alone. Because as we know, our HER2 does increase HER2 and uPAR does increase uPAR, but both together have an exponential increase, and that could lead to that chemo resistance. And just one highlighted figure from the paper was that when they looked at the expression, here they looked at a lot of different cell lines, and they found that most cell lines had a very strong correlation. If a cell line had a high amount of HER2, for example, this outlier right here, it also expressed a high amount of TLR, which is the gene symbol for uPAR. And that was very interesting because at the same time, the opposite of the spectrum, if there wasn't a lot of HER2, that means there wasn't a lot of uPAR. So it kind of shows this notion that HER2, that uPAR can be used as an indicator early on to whether this patient will respond well to HER2 target treatment, or if we have to look further for other or more complex kind of treatments in these specific patients. So next we'll take a dive into small lung cancer cell lines. And similarly to the HER2 study, this was also done on human cell lines where they took a bunch of different cell lines. And as you see, they treated them with known drugs that work on small cell lung cancers. And here they used cisplatin, etoposide, and a combination of both. And as you expect, the more you add of a drug that's been known to work, the less the viability of the cells, and that's predictable here as their control. But the interesting aspect and the interesting approach that they took was that they treated these similar cells with these drugs. But once they finished treatment and they took those remaining colonies that were still alive and they antibody fluorescent tagged them for uPAR. And what they found that the remaining cells after being treated were uPAR positive. So that kind of gave them that notion that uPAR might be playing a role in these resistance of the specific subset of these cancer cells that the classic methods of pharmacology are not working towards. And this approach is really interesting because it's very much a shotgun approach. There could be a number of proteins causing other effects, and the fact that they narrowed on uPAR is an interesting approach. But then they followed that up with a few more experiments, especially when it came to colony growth, where you can see here in just methylcellulose media, they just grew two types of small cell lung cancers, one with uPAR positive and one with a uPAR negative. And here you can clearly see the rate of growth between the two, the rate of growth that uPAR gives to these cell lung cancer. And these are not singular cells here. These are colony of cells. So I want to say a few millions in cell lines. So that just shows you another image of how uPAR is playing a huge role in this cancer's metastasis and growth specifically. And lastly, in prostate cancer, and this hasn't been done in human cells yet, but this one was done in prostate cancer cells and mouse cell lines. And this paper focused on MRI-143, which is a uPAR target actually, and they were hoping to show that MR143 targeted uPAR as a possible treatment for prostate cancer. But I just wanted to focus on the significance of cell death in these colonies, which I think are some beautiful images, and how SI knockout of uPAR has such a huge dramatic difference in the colony growth of these prostate cancer cell lines. And as you can see here, if you want an engraft method, it was hugely significant that when you knock down uPAR in these cell lines, you have a huge decrease in growth. So it kind of shows that there's a theme for uPAR functioning in these cancer cell lines. One, that it promotes very aggressive types of cell lines, and secondary, that it really provides a very ideal growth environment and in a really rapid manner. This paper went on later to show that MR does show slight significant decrease, but this was also seen here and it wasn't significant initially. And they kind of went on to say that, oh, their method of delivery wasn't perfect yet, so they're still working on it, but it just shows the potential of uPAR and the roles of cancer. So lastly, I feel like these days you really can't end a talk or study a protein without looking and diving a little bit into COVID-19. And this was a very recent publication, actually, not targeting uPAR in COVID-19, but moreover showing that the increase of uPAR in COVID patients had an increased risk of respiratory failure. So they wanted to use uPAR here as a marker. And what they found was that when the uPAR was increased, they hypothesized that this led to an increase in interleukins and integrins and that inflammatory state that damaged, that was so unpredictable from one person to the next in COVID-19. And the data was quite promising. So once they found that uPAR was high in these patients, so they took any patient with higher than 6 milligrams of uPAR in the blood, and then they treated these patients with anakinra, and they found that these patients responded really well compared to the similar patients who had a high level of uPAR that weren't getting treatment. So this is kind of an interesting aspect where uPAR might not be the target of the therapy, but more used as that indicator of what type of therapy we're doing. And this kind of similarly to the HER2 cancer earlier, where it's not essentially that we're targeting uPAR in this scenario, but uPAR gives us an indication of what type of treatments we might need down the line compared to the standard methods of treatments we use. And that kind of wraps up my little section. Mostafa will just be finishing us off with one more slide looking at future directions for this article and this wonderful protein.
Yeah, so just to sum it up, again, I find this protein very, very fascinating, and a 20-minute presentation doesn't do it justice for the extent that it's involved in the human body and biology and its application into medicine and in the clinical aspect of healthcare. So there are currently therapies that have been designed to target viral serpin proteins that have been under investigation as a conjugate towards binding uPAR and the uPAR complex. However, much more extensive research into specifically targeting uPAR in the bloodstream, for example, or as a monoclonal antibody or a chimeric antibody, whatever the form may be, is warranted and is needed to see, okay, does targeting uPAR actually provide a benefit to the patient? Is it a mortality benefit? Is it for example, heart remodeling benefit? Is it a longevity benefit to this to the patient? And specifically further delving deeper into, for example, can this be applied into chemotherapy? Can this be a conjugate to surgery and such? And that would be a very interesting research and field to see, and hopefully one I would like to see grow and flourish. Another possibility that this uPAR protein can take a course towards is being used as a diagnostic or a standard of diagnostic and prognostic tools. So we've seen research articles that have mentioned use uPAR as a way to measure the severity of coronary artery disease, for example, or the severity of stroke, and its ability to predict mortality for the patients. However, this is only in the research case. However, I would like to see a more extensive and expansive clinical trials, seeing can uPAR actually be used to detect, for example, a bedside at the bedside for a patient's capability of or probability of developing another heart attack once they had a PCI or once they already had an MI earlier that morning. So seeing that applied into a clinical trials would also be very interesting and provide us more insight into this applicability of this wonderful protein and dynamic protein. So I appreciate everyone listening to our presentation, and we will be here to answer any questions that you may have. And a quick shout out and thanks to, oh, these are the references actually, but a quick shout out and thanks to Dr. Lucas for allowing us to present this paper and providing us guidance in writing this paper and publishing it, as well as Kyle, who is a grad of KCU actually in 2017, sorry, 2021. And he also helped a lot with the editing of the manuscript and providing insight into this. So appreciate your time and thank you very much.